Today we are here to be witness for the final PhD seminar. We have two PhD seminars, and at first I will call Mr. Ikram Hussain. Mr. Ikram Hussain did his MSc in 2001 and MPhil in 2021, and now he is registered for the PhD program in 16 March 2023. He is serving as the Assistant Director in Gilgit Baltistan. His topic is the taxonomic and nutritive variegation of cold water fishes of Baltistan region and Devsai National Park, Gilgit Baltistan. So now I invite
Professor Dr. Nahid, Department of Chairman, Department of Fisheries and Aquatic Sciences, Dr. Khalid Rajnarisa, and respected Dr. Karim Gabul Sahab, Professor Dr. Karim Gabul Sahab, and other elite and esteemed professors and faculty members, and my dear students, Assalamu alaikum. It's my privilege to present today here in this office my final PhD seminar titled is Taxonomic and Nutritive Validation of Cold Water Fisheries of Baltistan region and Devsai National Park, Gilgit Baltistan of Pakistan. So, let's come to the contents. First, the review of the seminar, and the second one is the review of second seminar, then final seminar. To discuss the review of first seminar, I have done the two parts of my PhD dissertation research work. That is, the first one is the water quality analysis, and the second one is the GIS mapping of water resources and watershed typing. Water quality analysis, in this part, I have collected the seasonal variation, the physico-chemical characteristics along the seasonal variation, the parameters that I have studied, and they are collected through the standard procedure methods, that is the multi-parameters meters. And in this part of the research work, I have done the atmospheric temperature as well as the water temperature, the pH, dissolved oxygen turbidity, and so many other parameters. These are the water quality parameters of the desired water bodies, the sampling stations that I have studied in my PhD research topic. The first one is, these are the total 13 number of 16 number of sampling stations. I have selected 16 number of sampling stations, and the GIS-based mapping. GIS-based mapping, here I have selected 3 major rivers where the endemic cold water fisheries of fish fauna of Gilgit Baltistan are found in these rivers. So this is the Devsai Plateau, which is part of my site, research site, and the watershed delineation in this part I have collected, and I have 3 major rivers having a plenty of fish, endemic fish fauna of Gilgit Baltistan. The first one is the Shatong River, the Devsai National Park. The second one is the Bada Pani, and the third one is the Gultari River. In GIS mapping, these are some of the characteristics that I have studied in this part of the study. 3 major rivers, that is Shatong and Bada Pani and Devsai National Park and the Gultari rivers. Watershed delineation is also a part of my research study. And land use, land cover. Here is a slide. I have shown some of the drone imagery and LULC of the so many the Shatong River. LULC of the Shatong River. This is river shed detection of Chatham River. From 2000 to 2023, this overlay from the more than 20 years. These are the drone images of Bada Pani, and also these are the drone images of Guthri River. Now the review of second seminar. In second seminar, I have performed the morphometry of more than 11 indigenous fishes, and the second part of this seminar was the genetic analysis of 3 major endemic fish species. In morphometry, these are the sampling institutions which I have collected, which I have sampled more than 1,000 fish samples out of these 13 sampling institutions. This is the GIS map. And the sample collection, study duration is 14 July 2022, starting from the 2022 and the 15 October 2025. It ends. Fish sample, I have collected fish sample using billiard, cast net and so many other hooks and rods, the fishing methods. Preferred time, the preferred time for any sort of fish sampling is the early morning hours and the late evening hours. A total of 1,000 specimens belonging to 11 fish species were collected from 16 sampling sites, and the taxonomic studies on the site, mostly the taxonomic studies I have performed at the site, but sometimes due to weather or due to the time shortage, I have collected some of the samples preserved in the 10% formalin, then moved to the research lab of fisheries department of the district concerned. Identification of fishes, normally identification of species has gone through, these are the standard methods, standard keys, through morphometry experts, supervisors and local fishermen. Keys I have followed, these are the Jairam, Mirza and Sandhu, and the literature and the internet sources from the gene bank. In a morphometry section, there are 27 morphometric characteristics I have studied, and these are the characteristics that I have studied: the body weight up to the ventral lateral distance. Meristic characters has always been documented in this morphometric section. This is the meristic count, and the meristic count having more than 10 traits have been studied so far. And the fin formula is that the dorsal fin, caudal fin, and pelvic fin. These are some of the stock identifications and the morphometric analysis which we have studied in the field. And this text has been taken in 8 July 2023. These are the formalin preparation and field for field study in their side plans. And these are the morphometry of their side fishes, morphometry of Indus River Sigardo range, morphometry of Shiok River, and these are the Kapulu confluence of Indus River and Shiok River. So I come to the result of the second seminar. The first part I will discuss the morphometry. The morphometry of more than 11 species have been done in this seminar. The fish species and relative abundance from the study zone. These are the fishes and their relative abundance, that how much these species diversity has been found in this particular study. So the first one is the Schizothorax, Schizopyropsis toliki is the fish that have mostly found in the major rivers of Gilgit Baltistan, and it is the 32% total out of the total. And the lowest is the Schizothorax associnus, which is hardly the 1%. Morphometric trait, the species of Schizopyropsis toliki, these are the morphometric traits I have studied. And Diptychus maculatus, Tychobarbus chondritrosus is another fish, and Indus snow trout, Himalayan snow trout, and show xenophorex. These are the species that I have studied in the morphometric analysis. So, in this, the second review seminar, the second seminar, I have already performed the genetic analysis of 3 number of fishes. These are the 3 number of fishes that we have collected from their side, and the 3 major rivers from Shatong River and the Guttari and Bada Pani. Our major components of genetic analysis is NCBI online blast results, gene pole expression and mitochondrial and DNA expressions, and scientific illustration and other informations. NCBI online blast results. I have performed the sequences and the DNA, the PCR and all other related procedures from the concerned lab, and then I have got the sequence data, and then I have blast these data to the NCBI online platform. So this one, the species that I have done for the sequence analysis is the Diptychus maculatus, and it shows the 98% identically, and expression is QD cover is 97%. So with the help of the morphometric result, which resemble the 100% resemblance to this species, then so for this result we have selected that this species is the Diptychus maculatus, and the mitochondrial genome notation of that species, and this is the species that the Diptychus maculatus. We have classified and updated the result, validated from the genome sequences. This species is also known as Scaliosman, and the major tributary is the Devsai and the Shatong River. Phytobarbus conirostris is another fish. This is known as Indus snow trout, and this is the endemic fish of Gilgit Baltistan. So this species has also performed the genetic sequences, and the NCBI blast I have seen here, gene pole expression, gene bank accession number, and for this species I have got the accession number as well. I have the sequence data I have submitted to the gene bank, and the gene bank has and I successfully availed the accession number. This is the first ever accession number any kind of fish species in Gilgit Baltistan. This is the Phytobarbus, is found in the Devsai National Park. And CBI blast, this is the Triplophysa loge. This is the third one species is the Triplophysa loge. In Triplophysa, this is also, this species is also validated. I want to discuss something about this Triplophysa loge. So Triplophysa loge initially in early recorded, some of the scientists they are called this species is the Triplophysa loge, and the scientific name is Triplophysa stoliki. But in our validation process, we have classified that this is not the Triplophysa stoliki. This is genuinely the Barbatula barbatula, and this is the new record in the history of Gilgit Baltistan. So the third one is the Barbatula barbatula. This is the Barbatula barbatula that we have collected from the Guttari River. It is also commonly known as the stone lodge. And here I come to my final seminar. The content of my final seminar is the introduction, objectives, material method, results, discussion, conclusion, future work, references and acknowledgement. Introduction, as far as concerned with the Rafiq and Khan, they have studied the freshwater fishes of Pakistan. There are 193 fish species recorded, and Pakistan, out of the 193 species, these are the Pakistan comprises of 190 and 3 allies, 5 superorders, 10 orders and 26 families and 82 genuses. Cold water fishes are Pakistan. These are the 28 cold water bodies in Pakistan, mainly confined to the Trans-Himalayan Indus River system. And the cold water active fauna is mainly, particularly these are the Shirothorax, Diptychus and Dicobarblus. The fish fauna is Gilgit Baltistan. Pakistan, the Gilgit Baltistan is a mountainous region and covering the cold water fish fauna of Pakistan. And predominantly there are 17 fish species have been recorded in Gilgit Baltistan. And the second part of my study is also the nutritive validation of that particular fish species that I was studied in my PhD dissertation. The nutrition analysis, these are the four components that are called the proximate analysis, and these are the moisture, crude protein, crude fat, and the ash. And so many other nutrients I have also studied, that are the carbohydrate and energy value as well. In any sort of fish body, the water is 70% crude protein, 18%, and crude fat is 0.5%, and mineral is 0.5 to 15% as well. Other mineral components are also in the fish body. So this is my specific objectives of overall my PhD topic. Develop GIS mapping of the study area, water quality analysis, study morphological and morphological and basic characterization, taxonomic validation through DNA-based molecular methods, examine the evaluate nutrition composition of cold water fishes of Pakistan. So now come to the material method topic. Material method, in material method I have sectioned these 5 categories: study area, GIS mapping, sample collection, nutrition analysis, genetic analysis, and statistical analysis. These are the description of the study. Throughout my this PhD journey, the topic is the habitat and all the river system. The study area I have categorized in 2 core zones. The one is the Delhi National Park, with the second highest plateau in the world, and the second one is the Baltistan region. In Baltistan region, these are some of the rivers: Indus River, District Sikardor Range, Brahim Lake, Shiok River, and District Galge. All sorts of 16 sampling restrictions have been studied and have been selected in these 2 core zones. The GPS coordinates and elevation and annual water temperature have been recorded so far. GIS mapping of the study area, whole of the study area. This is the GIS map, and Hartman range, different sampling restrictions. This is the GIS map of Sikardor range, Indus River. This is the Shiok River, Ganchi district. And these are the map of the Delhi National Park. Fish sample collection and processing. These are the basic methods, basic standard methods that I have collected the fish samples and for further analysis as well. Methodology for nutrition analysis. The methodology is that each and every sample that we have collected, for the recommended weight of the sample should be 250 gram. Analysis, the nutrition analysis of the fishes, and the nutritional fish sample at PCCR Karachi. The second part of my final PhD seminar is the genetic analysis. In genetic analysis, the methodology, I will discuss the methodology, sample collection for the genetic analysis. Normally for genetic analysis, I have collected the sample of the caudal fin or the meat sample, as well as sometimes I have collected the blood sample as well. At the cleaning section of fin clip fish flesh, weighted up to 5 gram, and for further DNA extraction process has been carried out in the concerned laboratory protocols. And the flesh sample first descaled and filleted annually, manually using sterile plastic knife, and so many other standard protocols. And transported, all these samples have been transported via airplane to Alpha Genomic Lab Islamabad, where these are the standard analysis have been carried out. The sample labeling is also an important task of the sample collection methodology, in which we have written the name of the sample with the species name, individual code, location, and sampling institution name, GPS coordinates, altitude of the sampling site, data, date, and the name of the researcher. Sample processing for molecular genetics, these are the validation or the genetic protocol, the DNA extraction, then PCR amplification, thermal cycling condition, and initial denaturation, and the final, the extension, PCR product when analyzed, 1.5 aggregate gels, and then sequences have been collected from the third party as well, or from the lab authorities. Then these sequence data have been accessed to the NCBI BLAST, and accession number were obtained for confirmed taxa from the gene bank as well. These are the mitochondrial assembly and integration. This is a methodology and the workflow of the sequence data for constructing files. These are the some of the glimpses that during the genetic analysis, the field survey, we have taken throughout in the Baltistan region, the study area, Baltistan region, and in the Baltistan National Park. This is the Baltistan National Park, where we have performed the genetic analysis, tagging, the sampling, and all other related activities for further genetic analysis. These are the some of the weighing, degating, and tagging of the nutritional samples, and preservation and tagging for genetic analysis. The Bada Pani in the Baltistan National Park, and this picture has been taken in 11 July 2023. These are the collection of blood samples. As here we are saying that from the caudal fin we have collected from the blood for validation of fish species, and this is the species that we have already discussed in the second seminar. Newly recorded fish species Hilly Stream Loach, and the scientific name of that fish is the barbatula as well. Collection of blood sample for recorded fish species. So now I come to the result of my final PhD seminar. The result I have categorized into two parts. First one is the nutrition analysis, and the second one is the genetic analysis. So in the nutrition analysis, as I already told, that the proximate composition, heavy metal, and the Mendel profilation of 11 number of fish species, more than 11 number of fish species have been taken, and they performed through PCSR laboratories. And the second one is the third one in the Mendel profilation, and they compare with the data, all the data with the WHO permissible limits. Comparative proximate composition energy value of Tychobarbus. These are the two ecotypes that we have compared from the one I have collected from the Desai, more than 4,000 meters above sea level, the altitude is, and one of that species I have collected more than 2,000 altitude from the Shiog River. So we have just in this particular part we have studied the effect of altitude, the effect of temperature on the proximate composition of the particular species. So these are the proximate compositions that I have studied so far. The first one is the barbatula, or these are the nutrient scale, the nutrient percentages, and this last one is the schizothorax paleostomus. And here one thing I have told that the effect of altitude is that when we go through the altitude and the temperature regimes, so one thing we have discovered that when the temperature rises and when the altitude rises, then the fat deposition is also the rises. So for this reason, automatically protein percentage is lowest as compared to the highest zones. Proximate composition, heavy metal profilation. In this case, I have studied the heavy metal detection in so many fish species, and there is no, the data is under the permissible limit is recommended by the WHO and the FAO. These are the comparison of heavy metal with the WHO and FAO permissible limits, and here I have circled one of the salvotropoferio, the figure of the selenium. It is within the safe limit, but it is near the limits. So this one is quite alarming. This data is collected from the salmon tropoferio, that is also commonly called the brown trout as well. This is the Mendel profilation. Mendel profilation is also under the permissible limits, and here the permissible limits I have already mentioned, the FAO and WHO recommended safe range, and all the fish, the minerals contained in the selected fish samples have under the permissible limits. Comparative proximate comes to energy value of trichobarbus that I already mentioned that these are the two ecotypes that are collected from the ecologically diverse regions. First one is the elevation is 2500, and the second one I have collected Chatham River their side. Both these are species, the same species, but I collected from different elevations and different temperatures. So one thing that I have mentioned already, that in Shiok River, you see the fat level is 2.1, and in the Chatham River, which is high altitude and the lowest temperature, the fat level is the 4.19. So the second part of my final PhD seminar is the genetic analysis result. In this part, I have studied more than 9 fish samples have been sequencing so far. And the second part, I have compared the current finding with the previous scientific data. Detail of accession number from the gene bank. So the first, the fish that we have already accessed the accession number from the gene bank, international gene bank, that is the barbatula, and these are the accession number TQ35 and 4418. And this is collected from the Gultari River. Village name is the Frenchard, Siping, Sikabu, Gilgit Baltistan, Pakistan. The second species, that is the Tychobarbus conirostris. It is also called in the snow trout. And that species that we have also gained the accession number from the international gene bank. The third one species is the Diptychus maculatus scariosmum, and it is also locally called Dersidia also. Then Sibai blast result card. This species, the first time we have validated through whole genome sequence method, and this species is now we have validated is Diptychus maculatus. The gene pool expression of this gene bank submission report, and the Galactostorum maculatum. This is the fourth species that we have submitted, and the gene bank have accepted our data, whole genome sequence. Galactostorum maculatum is also called the Himalayan catfish as well. The SCBA blast result card of this species is Galactostorum maculatum. Previously, this species have been misidentified as Galactostorum reticulatum. But after this, after our work, we have validated this species as the Galactostorum maculatum. The gene pool expression, and the fifth species is the Chirothorax radiatus. This is also the Tunas snow trout, and the whole genome sequence data is already submitted to the gene bank authorities, and they have accepted. And this species is previously reported as Chirothorax indicus, and now we have confirmed that this species is the Chirothorax radiatus. This is the blast result card. And the sixth species is the Tychobarbus conirostris in the Stoco. This is the riverine form. We are collected from the Chatham River. We have collected from the Shiok River. And the gene pool data annotation. The seventh species is Pseudopaigopsis sturicai. This is locally called as Ladakhi snow trout. But now we have validated this species is the Tychobarbus conirostris as well. The fifth one is Ladakhi snow trout is also in the previous literature. This species is also misidentified as Schizopaigi sturicai, but now we have validated this species Schizopaigopsis sturicai. Kunar snow trout Khachuga is also known as locally known as Khachuga. So the sixth species we have also validated. Schizothorax intermediate. Earlier reported this species is Schizothorax intermediate, but after the genome sequences, whole genome sequences, after the SBI blast and the standard procedure, now the current finding is, and the species name is Schizothorax labiatus. Himalayan catfish, the seventh one, and locally called as Zoko. This is also called Himalayan catfish. In previous literature, this species is also called Gleptosternum reticulatum, but now this species is known as the Gleptosternum maculatum. Himalayan snow trout, Recoma labiata. This species is also mislabeled and misreported is Recoma labiata. But after the validation process, but after the genome sequences and the CBI blast and other standard procedures, the current finding is that this species is basically Schizothorax plagiostomos as well. Ninth one is the Scaliosmum. This is also a loach, stone loach, commonly called as the stone loach. And earlier, the scientists data this species is Triplophysa yasinensis. But now, after the gene bank accession records and the NCBI blast, this species is also the Diptychus maculatus. And the fourth one, 10th one is the Chira snow trout, that is also known as Schizothorax assassinus. This is the same species that we have recorded. Discussion. Finally, I come to the discussion. As I have already told that. In the final PhD seminar, I have categorized all of the studies in 5 parts. That is, the first one is the water quality of the desired river sampling restrictions. Second part is the GIS mapping. Third part is the morphometry of the desired fish species. Fifth part is the genetic analysis, and the sixth part is the nutrition analysis. So these are the finding. The discussion is that all the water temperature, dissolved oxygen, pH, and other physico-chemical characteristics of the water bodies that are studied under the study area is suitable for cold water indigenous fish fauna as well as the exotic fish fauna, that is the commercially exotic trout, that is Rambo and Bronto. The current research work delivers sample assessment of the nutritional and heavy metal and mineral profilation as well, the nutritional analysis of some desired indigenous fish species, and while displaying the minor and specific variation poses a notable and load and dietary nutrient composition as well. Among the understudied that I have mentioned that more than 10 species they have undergone to the nutrition analysis procedure. And among the understudied fish species, the Tychobarbus conirostris and both the trout species followed by snow trout are the best in terms of protein value. And among various species of snow trout, the Indus snow trout, that is also known as Tychobarbus conirostris, and it is collected from the Shiok River, is identified as superior fish species with respect to protein content as well. The Himalayan snow trout, that is known as, the scientific name is Schizothorax plagiostomos, the second best just after the Tychobarbus conirostris, and the Ladakhi snow trout, that is also called the scientifically called Schizopaigopsis sturicai, the third best in one among the studied fish samples. As far as concerned, the level of the heavy metal, namely iron, mercury, lead, arsenic compounds, were under the permissible limits as recommended by the WHO. and other guidelines confirming the safety of consuming fish from the study area. The present study confirmed the continued presence of major cold water fish taxa previously noted from the Himalayan industrial system, showing overall taxonomic consistency with earlier literature. However, the result indicate taxonomic version updated nomenclature in several fish species that I have already discussed in my previous slide, such as the study validate the occurrence of large species and the Piprophysis stolicae is basically the Barbatula, indicating improved specific level identification. Some species is also reported under a synonym names such as Sephoma labiata are now recognized under the updated accepted name like Schizothorax pelagiostromus, demonstrating refinement in fish taxonomy. Galactosternum reticulatum maculatum is earlier reported as Galactosternum reticulatum as well. As far as the Tychobarbus conirostris, riverine form resolving historical misclassification as Schizothorax cicatricensis. This species normally in the old data, this species is called is normally known and reported as the Schizothorax cicatricensis, but now the confirmed after the gene bank validated data, this species is now the Tychobarbus conirostris. The present data reveal that the Schizothorax associations is a rare and habitat restricted species, aquatic system of Gilgit Baltistan with limited ability to confine waters of Glen Lake and lower stretches of Indus and Shyok River. Overall, the present findings support earlier record but provide more precise and scientifically updated species classification as well. The conclusion of the final seminar, my topic is that this research removed long-standing taxonomic confusions, validated species at molecular level for the first time, established nutritional and mineral benchmark for the cold water fisheries, especially the Gilgit Baltistan region. This work is a scientific milestone for GB fisheries in particular and in Pakistan in general. Recommendation and future work establish there should be establish the gene Gilgit Baltistan or more importantly the Pakistan fish gene data bank, and also develop. It's also recommended that there should be developed region specific nutrition data on the basis of the nutrition enhancement the particular region. Use validated data for species conservation, and these data we can use for species conservation and management, particularly those species we are in the hands of endangered and endemic species as well. Promote research collaboration in advanced laboratories for genetic validation and nutrition analysis for the remaining 8 indigenous species out of 17 recorded species. Here in this study, we have already performed the 9 species. Then it is highly recommended that remaining 8 species should be validated. An acknowledgement. I am thankful to Allah Almighty who gave me power to cover this task. Heartfelt thanks to my respected supervisor, Dr. Yunus Lagari, and my co-supervisor, Dr. Kunal Khan Lashari, for their support throughout my research and throughout my journey. I am very indebted to the Director of Fisheries, Gilgit Baltistan, and for their unlimited support during field visit and data processing. I am thankful to the Chairman, Department of Fisheries and Aquatic Sciences, Professor Dr. Khalid Ashari sir, for his moral support and care and each and everything. I am grateful to Dean Faculty of Science, ma'am, for your precious time. Special thanks to the Department of Fisheries, students of Department of Fisheries and my colleagues. These are Dr. Yunus Lagari sir, Dr. Kunal Ashari sir, from 9 to 14 July, that more than 1 week we have collected most of the data in this specific time. Thank you so much.
Thank you. Now the session is open for the question and answers. Please, if any. Okay.
Actually, I am not asking a question, but I would like to appreciate the researcher and able guidance of their supervisors who have done really excellent job, and the presentation was excellent and it was full of heavy words which were very difficult for a social scientist to understand its deep meaning. It is up to the external and the dean, they will accept it inshallah. But I would like, being a social scientist, I would like to would like to have seen a social aspect of this research, what kind of social benefits it would have for the people living in that particular area. So if you have anything to add or not.
Yes, sir, of course, sir. There are so many particular to the cold water fisheries. You know, sir, that this is the Gilgit Baltistan is quite a poor region. Most of the people, they are living in the hard hilly areas. Especially, they are doing the fishing and for their livelihood development process. And second thing is that with particular reference to social ethics, we also add that this is also a management and conservation part of that particular fisheries and aquaculture activities.
Great, thank you, sir.
You know, so your presentation was wonderfully well. My question is this: having the biology background and pursuing my MPhil in that particular department of fisheries, I'm also particularly interested in, you know, genomic sequence and their, you know, this kind of, you know, that's kind of my interest. First of all, my humble question here is that, you know, you perform the genomic sequence of the 9 fishes. That is extremely wonderful work. But my question is, did you do it your own self by the first-hand practice, or it was just the, you know, lab work from somebody else that they were performing and you just use the collected data or just the analysis report? I mean, you did it by your own self, or it was just the analysis report from the lab?
Okay, thank you so much for this very nice question. Actually, more than 90% of the work, which is all the procedure and the methodologies that are available in Pakistan, we have done with ourselves.
Yeah.
That is mostly the nutritional analysis we have carried out from the PCR laboratories. And the second one is that there is some sort of difficulties we have faced in genetic analysis.
Yeah.
In Pakistan, the sequencing has been performed. So the sequencing procedure we have availed from third party, from the foreign, from Japan and any other laboratory. Then we import the data and then we have gone through the blast procedure. But the samples, collection of the sample, preservations and the DNA, the PCR, each and everything that have performed in the laboratories of the particular lab which are available in Pakistan.
And I have like further question, the remaining fishes that you have analyzed like 9, but the remaining species of fishes that you have collected, they are available for further analysis. If I would like to work on that, is it available?
Yes, you are most welcome. If you are work the remaining 8 fish species, then as a part of from that I am from the Gilgit Baltistan, we also offer for you the services from the fisheries department Gilgit Baltistan. We have requested the fisheries department authorities that they support you, and I think you will do the remaining 8 species as well.
I once again appreciate, you know. Okay, but thank you for that.
Genetics. Genetics and recorded new species. You have collected sample from Gilgit Baltistan, Ikram the Gilgit Baltistan, and your work is true commendables. Okay. I personally witnessed with you when and was present there during the sampling process, and I hope your research work will play an important role in the Gilgit Baltistan's fisheries sectors and will serve guidance future researchers. I appreciate you and I congratulate you. You have done this kind of work. Thank you.
Thank you, sir. I also, sir, I'm really privileged, sir, that you also being a part of that research studies, sir, who validating as far as through the genomic process as well. After this, Dr. Rafiq, sir, and I'm privileged to work with him for 2 years as well. And he is also the known taxonomist as well. So the difference is that, ma'am, that in this particular study, we emphasize, we focus the validation process through the standard genomic procedures. This, and additionally, in addition to this, we have just collected the nutrition data.
Tell us about there are 9 species you identify.
Exactly, ma'am.
On the basis of?
On the basis of, ma'am, both from the morphometric and the genetic analysis, that is sequencing, the phdvlogs, ma'am. That's
Morphometric, which type of E you have to use?
ma'am, sometimes that we have my honorable supervisor and co-supervisor, we have discussed a lot. Some of the species, just for example, ma'am, some of the species that is.
Random questions, what the key you use for the identification?
Name the key, only one word.
No.
Then
You referred 3 keys in your material.
No. Okay, the level is mine. There is the carnivorous diet as well, and there's some sort of because they have feed on mostly the zooplankton, and for this reason. And the second one is the rainbow trout. Rainbow trout is basically escaped from the fish farms as well to the wild origins.
okay, over to experts.